circulation and then embryology. Single versus double circulation, just talking about the pulmonary and systemic circulation. So remember that basically in things that have a double circulation, this is not drawing, but you can just see schematically that with a double circulation, you basically have two circuits. So one is the lungs and the other is the body. And if you have a single circulation, then you just have like, one circuit. Usually going to the lungs. Okay, so this is what's meant by a single versus a double circulation. So in the single circulation, blood is leaving the heart, going directly to the gills, and then to the body, and then back. In the double circulation, it has two circuits. So it goes from the heart to the lungs, and then from the heart to the body. Two separate circuits. In fishes, there's a single circulation. So again, blood is passing through the heart only once. Interesting fact that you probably did not learn in intro bio is that fishes actually have a four-chambered heart. But it's not the four chambers that we talk about. So the four chambers in a fish are the sinus venosus, atrium ventricle, and the conus arteriosus. So think of the shark. In the shark, the sinus venosus opens into the atrium and then the ventricle, and then the conus arteriosus, which goes to the ventral aorta. So these are actually chambers in a fish heart. And what happens when we talk about tetrapods, in contrast, is just that we double the atria and ventricle, and now the sinus venosus and conus arteriosus simply become really parts of blood vessels. Okay, so amniotes, meaning reptiles, mammals, and birds, have this double circulation with blood passing through the heart twice. So once it goes on a short side trip to the lungs, and then the second time it goes around the whole body. There are many intermediates, like lungfishes and amphibians and some reptiles, in which there's actually both lungs and gills in the case of the lungfish. In amphibians, when they're in water and breathing with the skin, they actually don't need the lung circulation. So a frog, for example, doesn't require its lungs when it's under the water. It can just breathe with the skin. So I want to talk a little bit about embryology. Basically, the blood vessels in heart arise from the embryonic mesoderm. And what happens is it forms very, very early, almost immediately after formation of the mesoderm itself. Why? Any ideas why? So anyone want, yes?
Isn't it kind of like on the first testing, like to transport the nutrients the baby receives from, rather the fetus receives from the mother?
Good, yes, exactly. So it's to transport nutrients. The developing embryo requires nutrients. And whether it's in an egg or live birth situation, basically the developing embryo is going to require nutrients. So in the case of an egg, it's coming from the yolk. The circulatory system forms very, very early in order to transport nutrients to the developing embryo. So it starts with these tiny clusters of mesodermal cells that are called blood ions. These are the first things to form, and they look something like this. So here's the developing chick embryo. You can see this is going to be the embryo. And then remember that in the chick you have this flattened blastula and subsequent stages. So this is all still part of the embryo, and these are the blood islands. So blood islands are these little dark colored specks down here. And they are going to become both the blood vessels and also the blood cells. So they actually form together simultaneously. So basically, these blood islands form first and then subsequently become the circulatory system. And they're responsible for two processes. So we have hemopoiesis, which is basically formation of blood cells, which takes place even before the blood islands merge. And then the blood islands later will merge together to form tubes, and that's what we call angiogenesis, which is the formation of blood vessels. So the blood cells and the vessels actually form from the same tissue primordia, and they form at the same time, basically. Okay, so hemopoiesis and angiogenesis. The heart and the major vessels develop shortly after that in what's called the cardiogenic mesoderm. So that's part of the splanchnic mesoderm. Remember the interior layer of the lateral plate mesoderm right next to the endoderm. So that's going to become the heart. And the embryonic heart is tubular, and it very much resembles a fish heart. So we have in humans as well. So we have a sinus venosus, even in the human embryo. And then there's an atrium, a ventricle, and then what we call the bulbus cordis, which is actually a homologue of the conus arteriosus. So it's the same thing. And later on, it folds around itself to form the heart. When it first forms, the heart is already contractile. So remember, it has this these myogenic contractions. So the contraction, the muscle itself contracts on its own. It doesn't require nervous innervation. So as soon as these vessels begin to form, they're already contractile. So the heart begins beating pretty much as soon as it forms. And you can read about how it forms in your book. It's very interesting. It begins with these two tubes that fuse together. So you can read about that in your book. But I want to talk a little bit briefly about the phylogeny of the circulatory system. So this is an important thing because this is homology that goes all the way back to fishes. So primitively, vertebrates have 6 aortic arches. Think back to the shark that you dissected in lab. The aortic arches basically, you have the ventral aorta, and then afferent and efferent vessels going to the gills, and then the dorsal aorta bringing blood back. The same thing is happening in the amphioxus, pretty much, only with slightly different names. So in embryos of almost all gnathostomes, so most fishes that are jawed fishes and all tetrapods, the embryo primitively has 6 aortic arches, and then it gets modified through development. The adult pattern varies a lot among taxa, but all the embryos have this pattern pretty much. Aortic arches in reptiles, birds, and mammals, or in the amniotes, the most important ones are arches 3, 4, and 6, and we're going to see why. So what you should know is that arch number 6 is going to form the pulmonary trunk, pretty much beginning with lungfishes. And then we have arch 3, which forms the carotid arteries, and arch 4, which forms the systemic aorta and the dorsal aorta. So let's take a look at this. All right, so it's arches 3, 4, and 6. Keep that in mind. All right, so first of all, I'm just going to skip around a little bit. Here's our amphioxus, and you can see back to the first exam, and we talked about this. It doesn't have a heart, but it does have a sinus venosus. And the blood flows primitively, in amphioxus. It forms anteriorly along this ventral blood vessel, or ventral aorta, that's called the endostylar artery, and then it flows dorsally from there through these short blood vessels here, and then to the dorsal aorta on the top. And in the dorsal aorta, it flows backward. And this is a pattern that's very, very pretty much conserved among vertebrates. So this is Amphioxus, obviously not a vertebrate, it's just a chordate, protochordate, but in vertebrates this is conserved. Amphioxus has numerous aortic arches, not just 6, it has many, 20 or more. And some very primitive fishes that don't have jaws have as many as 10 to 12. So extinct ostracoderms, which are jawless bony fishes, have 10, and things like the lamprey has about 8. So the 6 sort of came a little bit later. So it's really a jawed fish or gnathostome adaptation. All right, so this is the basic structure. You can see these aortic arches here coming off this ventral aortic bone dorsally. Many, many more than 6. And then we can take a quick look at the shark, in which there are really only 6 now. And in fact, some of them get reduced. So you remember the shark from lab, so I'm not going to go over it too much, but you can see here, here's the ventral aorta, and now we have 1, 2, 3, 4, 5. The anterior one doesn't develop. So there's really 5 aortic arches in the shark. And then, but you can see looking at this that it's extremely similar. So just like Amphioxus, we have blood flowing anteriorly through the ventral aorta and then dorsally through these aortic arches, and then posteriorly through the dorsal aorta. And again, we have paired dorsal aorta in the head and then just a single unpaired one in the rest of the body. And then let's take a look at other vertebrates real quick. So here you can see some variation here. Teleost fishes really only have 4 aortic arches. The lungfish now has turned its swim bladder into the lung, as we mentioned last week. And it's interesting to note where this pulmonary trunk and pulmonary artery come from. So they're branching off of the 6th aortic arch, the efferent branch of the 6th aortic arch. So the pulmonary trunk comes from aortic arch number 6. And notice that this is called the ductus arteriosus here, which is connecting the pulmonary trunk to the aorta. So this is an embryonic feature in mammals. You can think of the fetal pig that you may have dissected in IntroBio, where you looked at the ductus arteriosus. It also connects the pulmonary trunk to the aorta. So even in humans, this is conserved, but it goes all the way back to lungfishes. Okay, so arch number 6 produces the pulmonary artery. Fast forward through evolution a little bit, you can see, so here's a larval salamander and an adult salamander. This larval salamander has gills, the adult doesn't. The larval salamander has a lung that it's not using yet, and then in the adult it's used so the gills disappear. In the adult salamander, arch number 3, aortic arch number 3, is going to become the carotid arteries. So carotid arteries develop from arch number 3. And in the larval salamander and the fishes, really it branches off of the dorsal aorta and the ventral aorta. So it's only in the tetrapods, including adult salamanders, that its arch number 3 really branches off to become the carotids. And now arch number 4, you can see here, in things like a frog, some of the arches disappear, and arch number 4 becomes the dorsal aorta. So arch number 4 is, the embryonic arch number 4 is what, the derivative of the dorsal aorta, basically. So you can study this in your book as well, but you can see there's this little thing in the middle called the carotid duct. between arches 3 and 4 here, and it just disappears in the adult. So this is found in embryos of amphibians, and then it disappears. So you can see it, in the embryo it resembles a fish, and then in the adult it's lost. So it's sort of ontogeny recapitulating phylogeny, or to put it in more simple terms, embryonic development, sort of paralleling phylogenetic development or evolution. Okay, we're pretty much out of time, so let's see. Yeah, there was a little bit more, so we'll talk about that on Thursday, and we're going to do digestion on Thursday. All right, so we're done. Any questions?